So next up we have Cécile, who's going to be talking to us about characterizing fine-scale SARS-CoV-2 transmission.
Yeah, hi. So I'm Cécile Tran Kiem. I'm a postdoc at the Hutchinson Cancer Center in Seattle. And I'll be talking about how we can leverage sequence data, more like specifically by using pairs of identical sequences to characterize disease transmission patterns. So pathogen typically spread through contact between infected and susceptible individuals, but because exposure and transmission events are rarely directly observed, understanding the factors that drive transmission risk is quite challenging. And pathogen genomes provide an interesting data source to understand where and how transmission is happening. But methods leveraging these data sources typically suffer from two limitations. So first, they're computationally very costly, so analysis have typically been limited to a couple 100, 2,000 genome sequences. And they've also been shown to be highly biased when sequencing is uneven. So we kind of really need new tools and methods to analyze large-scale pathogen genome data sets, such as those that have been generated during the pandemic. So over the course of transmission events, pathogen will accrue mutations. And so this means that we expect individuals who are close within a transmission chain to be infected by genetically closed viruses. And among these genetically closed viruses, if we're looking at identical sequences, these ones should be very informative about transmission events because we're actually really looking at the most epidemiologically linked individuals. And so in this work, we were willing to develop a method to look at where pairs of identical sequences are observed to characterize transmission from it. But one of the challenge we have is that the number of pair of identical sequences that we will observe, for example, in a geographic region, will be like highly dependent on the number of sequences that are coming from that region. And so to account for this, we introduced a measure of relative risk that quantifies how the number of pairs of identical sequences that we will observe in two regions, for example, so here red and green, differs from what we expect from the sequencing in this region. And so the relative risk here is equal to the ratio of two proportion among the pairs that are observed in region green, what are those that are also observed in region red. And we normalize this by the proportion of pairs observed in region red across all pairs of identical sequences. And so this makes this relative risk metric a sort of measure of enrichment with values greater than 1 corresponding to more pairs than what we expect from the sequencing effort. And so to apply these methods, we used a SARS-CoV-2 sequence dataset from Washington State with more than 114,000 sequences, for which we have detailed linked metadata that include age group and home location information. But looking at these sequences, we find that more than half of them have at least another identical sequence in the dataset, which means that we have a lot of pairs of identical sequences that we can look at to characterize transmission patterns. And if we're looking at a couple very large clusters of identical sequences that we observed here and like display them on a map, we find a strong both spatial and temporal signals. So for example, for this cluster here, with first some local spread around where it was first detected, and after a couple weeks observed at greater geographic distances. And so we applied our relative risk framework on this Washington dataset. And so first, we'll look at the relative risk of observing pairs of sequences within the same county. And so across all county, if we're looking at the relative risk of observing identical sequences within the same county, we find a median of 4.8, so corresponding to identical sequences having an increased risk of being observed within the same county compared to what we expect from where sequences are coming from. And when we're considering pairs of sequences that are situated at a greater genetic distances, the strength of that association kind of signal decreases to plateau at around 1. And so this suggests that individuals who are infected tend to transmit to people who live close to where they live, which might be expected, but it's good to know. So here we're looking at spread within the same county, but we can use this to look at spread between regions. And so here, for each of the county that is indicated by a red dot, I display the relative risk of observing identical sequences between this county and the other counties on the map. And so we see that relative risks tend to be high in counties that are geographically close. And as we're considering counties at greater geographic distance, the relative risk decreases. And if we're looking at trends across all pairs of counties, we find that the relative risk is greater within the same county than in adjacent county, than in non-adjacent county, which is overall like consistent with consistent signal for what we would expect under local SARS-CoV-2 spread. So next, we assessed how the relative risk of observing identical sequences compares with the relative risk of movement between regions. And so here we use two mobility data sources, so mobile phone mobility data obtained from the SafeGraph company, and commuting data that describe where individuals live and where they go to work. And we run generalized additive models to determine what is the percentage of the variance in the genetic data that is explained by mobility data. And so at the county level, we found that mobile phone and commuting data explain 63% and 74% of the variance in the genetic data. But at this county level, some of these relative risks are computed based on a very low number of pairs of identical sequences, and so we expect them to be more noisy. And so we also repeated the analysis by aggregating counties, and at this regional level, we find that these mobility indicators explain 80% of the variance in the genetic data. And I think also interestingly, these two indicators, regardless of the spatial scale, explain more variance than a distance-based metric only. So overall, this shows that mobility is a strong predictor of the location of pairs of identical sequences. So next, we use this generalized additive model that we run here to identify outliers in the relationship between genetic and mobility data. And so we define outliers as pairs of counties for which the absolute values of the residuals we obtained from the generalized additive models were greater than 3. And because we expect relative risk computed based on a low number of pairs of identical sequences to be more noisy, we only focused on pairs of counties for which we observed at least 100 pairs of identical sequences. And so we identified 2 pairs of non-adjacent counties for which the number of pairs of identical sequences that we observed is greater than what we expect from the relationship between genetic and mobility data. And so these 2 counties are, well, you've probably never heard of those, Walla Walla and Mason County and Franklin and Mason County. And so from like a demographic, geographic perspective, there's really like nothing very obvious that would appear to like, could explain these potential spread patterns. And so digging into this, we identified that these 3 counties were all the home of a male state prison. And so next, we investigated whether these outliers could be explained by SARS-CoV-2 transmission between prison facilities. And so to do so, we looked at spread at the postal code level. So here I depict the relative risk of observing identical sequences between Mason and Franklin postal code and Mason and Walla Walla postal code. And so we see that for most of these pairs of postal code, we never observe any pairs of identical sequences, most of the time corresponding to these 0 values here. But then every time this strong signal can really be narrowed down to 1 pair of postal code and to the same postal code in Mason County. And so in these 3 postal codes correspond to the postal code of the male prison we identified. So overall, this showed that these outliers are consistent with transmission between prisons during the pandemic in Washington State. We discussed these results with people at the Washington State Department of Corrections, who mentioned that during the pandemic, there's been movement of both incarcerated individual and staff between this prison that could explain these patterns. I think overall, this kind of exemplifies how, by combining genetic and mobility data, we can identify transmission pathways that might have been like under-recognized and that differ from the general community. So here we've looked at like spread between geographic region, but we can really use this framework to look at spread between any types of groups. And so next, we looked at spread between age group. And so here I depict the relative risk of observing identical sequences between the 0 to 9-year-old and other age group, and same thing for the 80+. So we see that in the children aged 0 to 9, the relative risk is highest with children aged 0 to 9, and it then decreases in other group with a little bump in the 30 to 49, so consistent with transmission happening between children and the parent generation. And when we're looking at the 80+, it's a bit of a different pattern with only a single spike in transmission between elderly individuals. And when we compare these 2 expectations from social contact data, we find that social contact data explain most of the variance in the genetic data. So here we'll look at like spread between region and age group kind of independently, but here for the very vast majority of sequences, we actually have that information available jointly. So we can use this to kind of understand how transmission between age group might be like mediated by a spatial scale. And so this is what we do here by computing the relative risk of observing identical sequences between age group, either by looking at all pairs of identical sequences, which is what I've just shown before, or only pairs in different postal codes and counties. And so for example, if we're looking at the 80+, we see that the strong enrichment that we had when looking at between LLI pairs completely disappears when we're looking at pairs that are more distant, which suggests that these types of transmission events tend to rather occur locally. We see the same type of decrease when when looking at between children pairs, which suggests that type transmission between children rather tends to occur at the local level. I think interestingly as well, if we're looking at pairs shared between children and elderly individual, we see the inverse pattern where the relative risks tend to be higher when we're considering more distant pairs, and so suggesting that these type of transmission events are rather associated with some kind of travels. And so I think this kind of suggests that this is kind of a first step, but potentially leveraging these mobility data for which we have both age and spatial information can help understand how social contact might vary and mobility might interact, which has been like pretty difficult to quantify because typically these data sources are gathered separately. So to conclude, we're using identical sequences, and we show this can provide insights into fine-scale transmission pattern. And here we're really relying on a simple count-based metric that should be easily scalable to large data set. And this is, there's a bunch of different directions, such as how to better in asymmetry and transmission between groups or estimate the contribution of different groups to epidemic growth. And so that will be it for this. I'll take the opportunity to thank all the people who contributed to that work, especially everyone who actually performed the sequencing and the linking of the metadata, and everyone more on the analysis side, including the other Cécile who's here. Thank you.
Time for a few quick questions. Yeah, look.
Thank you very much. You chose zero difference between the two, but that's basically you chose a cutoff of zero.
Mm-hmm.
Have you go to try to do one difference?
Yeah, so we've looked at one mutation. I think what's interesting is that so I think it's like kind of it would be a way if you didn't have enough pairs to kind of like inflate your data set. I think what we saw here though, what's interesting is that when we're looking at pairs one mutation away, for example, we no longer see the signal for transmission between children and parents. So I think it would work for, it wouldn't probably work as well for SARS-CoV-2 for a more subtle transmission signal. But I think, for example, here one mutation away, we still had the signal for like local spread.
Yes.
Yeah, so it's interesting that basically, maybe I'm misunderstanding, but is this sort of suggesting the opportunity to use this to update these both mobility matrices and age contact matrices? And so because of that intensive sampling that you need to get these pairs, do you think it's worth the investment of concentrating on specific pathogens for each route instead of having sparse?
Yeah. I mean, I think it's kind of like definitely the hope, and also being able to like, I think also like the idea of like this interaction, for example, between age and space. I think methodologically though, we're still pretty far. Like here we kind of like have a measure of enrichment, but we're kind of like, we're not at a contact matrix yet. And like the relative risk is impacted by sampling, it's impacted by the reproduction number. And so there's kind of like, I think it's like definitely something I'd like to pursue, but I think there's many steps to get there.
Okay, thanks very much, Cécile.